Welcome to this instructional video on the levels of protein structure. The first level of protein structure is called primary structure. It is the linear sequence of amino acids in a peptide or a protein. And we know that amino acids polymerize to make these peptides and proteins through what we call a peptide bond. And a peptide bond is really an amide. So if we look at an amide group, or in this case a peptide bond, let's put a little orange dot on this. Then I'm going to go down here and we have a stretch of amino acids linked via peptide bonds below. And I'm going to put an orange dot by each of the peptide bonds. One thing that we know about the amide linkage and the peptide, or in this case the peptide bond, the bond indicated by the orange dot, is that due to resonance, it has partial double bond character. You cannot rotate around the peptide bond. That means that you're going to have molecules fixed in plane with one another. And that's going to pretty much limit the structural possibilities of a peptide and a protein to certain specific types of structure. Now, before we get to that, let's make one other important point, is that the backbone along a peptide and a protein has direction. You may say, well, how do you know which ones are the backbone atoms? Well, any atom that's not in the side chain, and in this case, the R group, which is a purple sphere here, everything not in that is going to be a backbone atom. And what we know is that many of these atoms in the backbone are going to be in the same plane. Now, in terms of direction, there's always an amino terminus and a carboxyl terminus. Okay, and we're typically going to be moving from amino to carboxyl. And how can you figure out which way we're going? Well, we'll look at this. So here's an alpha carbon, here's a carbonyl carbon, there's a nitrogen, again, of the amide linkage. Here's how I remember this. This nitrogen came from an amino group. It's not an amino group anymore, it's an amido group, if you will, an amide. But it came from an amine. So typically I say amino alpha carbonyl. Amino alpha carbonyl. Amino alpha carbonyl, amino alpha carbonyl, amino alpha carbonyl. You can keep going. So I know I'm going from left to right here, the amino terminal end to the carboxyl terminal end of the peptide or protein. Okay? So that's what I mean by the backbone has direction. Now back to this planar issue. If you actually identify the atoms that are coplanar, you can actually see the plane fairly nicely. So along the backbone, the fact that the peptide bond has partial double bond character forces the 6 atoms involved in each one of these planes to be coplanar. And guess what connects the planes? Well, here we go. Alpha carbons. So the pink are alpha carbons. Now, to help us understand the next level of structure, we want to talk about the spatial relationship of the alpha carbons. Now, in terms of the backbone, these alpha carbons can assume different positions with respect to one another because there are 2 main bonds that can rotate along the backbone of a peptide or protein. And that happens to be the bonds on either side of the alpha carbon. So let's make this alpha carbon pink. This bond here can rotate. This bond here can rotate. This is why the backbone can assume different structural motifs or structural possibilities. because those two bonds can rotate. Any of the other bonds around, and any of the other bonds in the backbone will not be able to rotate because they are coplanar. Why are they coplanar? Because the peptide bond has partial double bond character. So, we've talked about primary structure. We reviewed the backbone a little bit. So, primary structure, what is the linear sequence of amino acids? And by the way, that's going to be important. The actual sequence is important. If you start rearranging the amino acids in a different linear sequence, you will get a different peptide or protein. Okay? So let's talk about secondary structure. So I've already captured the backbone atoms here and the planes and the alpha carbons as these pink dots, big dots. We know that there are three major types of secondary structure, and this has to do with local spatial arrangement of the backbone atoms. Okay? And to demonstrate these to you, I'm going to kind of draw something for you that demonstrates the spatial arrangement of just the alpha carbons. We're not going to worry about anything else. We're just going to talk about the alpha carbons. Okay? Just to, hopefully to try to explain this a little bit better. So, the first type of secondary structure is called an alpha helix, and it is a coil. The backbone coils, and the side chains will come off of the coil. Okay? Well, if we look at this, let's see if we can draw this. If I can draw this. There we go. So this is kind of the coil. And then when we talk about where are the alpha carbons now, let's just talk about the alpha carbons. We have something that looks like that. So, ladies and gentlemen, this is the coil of the alpha helix. Again, this has to do with local spatial arrangement of backbone atoms. And to demonstrate that to you, I've shown you the spatial arrangement of the alpha carbons, and I have omitted everything else. This is called an alpha helix. Another spatial arrangement of alpha carbons comes in the fact that they can be in a kind of a zigzag pattern. So let's see if we can kind of sketch that. Well, let's do it this way. So this is kind of a zigzag pattern of alpha carbons. Now again, go back up to this image on the right, top right. I'm showing you where the alpha carbons are along the backbone. To get the 40,000 foot view of these structural arrangements, I'm just showing you where the alpha carbons are at with respect to one another. If they coil, it's an alpha helix. If they zigzag, you get what we call a beta strand. And then beta strands are used to make beta sheets. Okay? Now it's kind of easy to show a coil, but showing a zigzag is a little bit difficult. So sometimes we will convert this to just an arrowhead. And then this will be the amino terminal end on this side, and then the head of the arrow denotes the carboxyl terminal end. So the zigzag, a little bit harder to draw sometimes, so we just use an arrow with an arrowhead telling you where we're going. So look up here. Here's a beta sheet. You've got 3 beta strands. And so this right here, one arrow is one beta strand. And to get a beta sheet, you need 2 or more beta strands, or 2 or more arrows. All right. That's two types of secondary structure. Think about the spatial arrangement of alpha carbons. Coil, zigzag. There's one more, and it's just called a beta turn. So here we go. I've copied this same backbone rendering for you where I've showed you the planes. I'm showing you where the alpha carbons are in pink. And here, this is a little more simple because really the backbone simply just reverses direction. So where we're going with this is kind of that. So let me mark this up. This would be the, for example, this could be the amino terminal end. This could be carboxyl. And then where are the alpha carbons? Well, they're just along the turn. And so this really is kind of a, if you want to say this, 180-degree turn. You're going one direction and you basically make a U-turn and you're going back the other direction. Beta turns are going to be very important. Again, this is backbone atoms are making the beta turn. Backbone atoms are making the coil. Backbone atoms are making the zigzag. Okay? And so this is going to be important for the folding of proteins that are soluble in water. So we've got a coil, a zigzag, and we've got really just a turn, which is very close to what we would call 180-degree, I guess, maneuver. All right, a little bit deeper into protein structure. If we talk about something called, well, we've done primary structure, we've done secondary structure. What is tertiary structure? Tertiary structure is when you pack together secondary structural elements. And you can see the the cartoon of this. Well, you see the coil? Yeah, I think you see the coil. Let me get the little marker here maybe. Here's a coil. There's a strand of a sheet. And then you see turns. You can see where the backbone is making an abrupt turn. So there's all three types of secondary structure here, but they're not all splayed out. They're all packed together into a three-dimensional structure. So really the tertiary structure is the 3D or folded structure. The three-dimensional or folded structure of the protein. Again, what dictated this folded structure? It goes back to primary structure, the linear sequence of amino acids. Now, a few more terms to cover in this video. If you will notice, the protein showed here is one big molecule. Look at the green amino terminal, and you can trace it. I mean, we could start tracing this if we wanted to. It starts out with a coil. Here we go. We're coiling. Okay? And if you could trace that until you get to the blue dot, which is the carboxyl terminal end of the protein. Ladies and gentlemen, this is one protein molecule. But one protein molecule can have discrete areas of structure. And that's where something called a domain comes in. If you look at this protein here, is it one molecule? Yes. But are there two independent, discrete areas of structure in the protein? I say yes, and I've highlighted them with the pink dashed lines. This protein would have what we would call two domains, because there are two clearly discrete, independent areas of structure, even though this is just one protein molecule. All right, so tertiary structure, packed together secondary structural elements, is the 3D or folded structure of the protein. And again, I would be remiss if I didn't remind you that, why does this protein have this particular tertiary or 3D structure? Because it is tied to its biological function. One more thing about protein structure for this particular video. Sometimes a protein needs to be in a multimeric state. This being one protein molecule is not enough. It needs to be in a multimeric state, like a dimer or a trimer. So this cartoon does a pretty good job of showing you this. I hope you can look at this cartoon of a protein and say, oh, I see there are two monomers. And in this case, we're calling them monomers. These are one molecule, one protein molecule. How do you know that? Because there's an amino terminal. There's the amino terminal end. Here's the carboxyl terminal end. This is just one big molecule. Go down here on the second part of this. There's an amino terminal end and a carboxyl terminal end. So we would call this one molecule now a monomer. And if there's two monomers, two independent individual proteins or peptides, it's called a monomer. And then if you associate them together, they're going to be called a dimer. A dimer. And then what we have there, if you have a dimer, you are going to have what we call two subunits. And the number of subunits is just the number of monomers. Let's see if I can write that a little bit better. And so there are two subunits in a dimer. There are two monomers in a dimer. And in fact, if you look at one of the monomers that I've highlighted with the purple dashed line, this subunit has two domains because there's two independent areas of structure in one molecule. Those are called domains. But there are two monomers to make up this structure, so we would call this overall structure a dimer. This is called quaternary structure. Quaternary structure. Okay? So, you can think of a subunit as a monomer, and I've defined it a little bit differently down here. A subunit is an individual polypeptide in a multimer. These are really two definitions that speak to the same thing, right? You've got a subunit is a monomer, and that monomer is one particular molecule that's involved in a higher order structure, like a dimer if there's two monomers, a trimer if there's three monomers, okay? Again, what's holding the dimer together? A variety of non-covalent interactions, and I've just happened to show you one here. If there's a positive charge in one monomer and a negative charge in the other monomer, then probably this is going to be an ionic interaction holding together the dimer. So you can have non-covalent interactions holding together a dimer, or you can have covalent connections holding together a dimer, and that covalent connection would have to be a disulfide bond. That's also possible. So we have reviewed kind of an overall protein structure. Primary, linear sequence of amino acids. Secondary, spatial arrangement of the backbone. What arrangement of the alpha carbons do we have? Is it a coil? Is it a zigzag? Is it a 180-degree turn? Then you have secondary structural elements packing together to make tertiary structure. That's the overall three-dimensional structure of a protein. Some proteins, to do their job, have to associate with another molecule or more. And then if that happens, we would call the individual molecules monomers. If you have two monomers, you have a dimer, three a trimer, and so forth. And each monomer would be called a subunit. And so a dimer would have 2 subunits. We're going to review all this in class. I just want you to have this video to kind of get a good introduction to the levels of protein structure. Thanks.